Clear water blue signal or cell-based biosensor for PFOS detection. Hi everyone, this is Farisna. Our synthetic biology team has designed a cell-based biosensor for PFOS detection. So I'll start off with the introduction. PFOS or perfluorooctanosulfonic acid, which is one of the most persistent and harmful pollutants in our water systems. It originates from firefighting foams, industrial waste, and manufacturing runoff, and once introduced into the environment, it remains there for decades. It gets accumulated in the human body and is linked to cancers, immune suppression, reproductive harm, and liver toxicity. Despite its danger, detecting PFOS currently requires high-end instruments like LCMS, which are expensive, lab-bound, and require trained personnel. So our solution is different. It uses living cells, no any instrumentations, and no any reagents. Once engineered, the biosensor bacteria grow themselves and respond visibly to PFOS without the need for electricity or any lab infrastructures. So this makes our system suitable for decentralized environmental monitoring, aligning with the UN Sustainable Development Goal 6 for clean water and SDG 3 for public health protection. So the rest part is explained by Subheksha.
Thank you, Farisna. Hi, I'm Suvikta. Now I'm going to continue our SynBio solution. So we built a synthetic biology solution using E. coli engineered to detect PFOS. The system is encoded entirely on a plasmid. It expresses a PFOS binding protein, human liver fatty acid binding protein. When PFOS is present, this binding event activates an inducible promoter that triggers expression of beta lactosidase, an enzyme that cleaves X-col, producing a distinct blue color. So, this simple genetic circuit gives us a clear visible signal. A water sample turns blue if PFOS is present. The design is low cost, portable, and functional without electricity or lab equipment. Depending on the concentration of PFOS, the system can produce a visible signal in as little as 2 to 6 hours. It is also easy to construct using standard cloning and expression protocols, making it scalable and reproducible across labs or field sites. Now, for the slides will be explained by Sunita.
To build this biosensor, we first assembled our genetic part using Gibson assembly or seamless cloning technique. The plasmid includes a constitutive promoter to express the PFOS binding protein, a synthetic inducible promoter, and a laxate reporter. We would use the standard blue-white screening cloning and expression in competent E. coli DH5 alpha as the host, culturing it under standard conditions in LB with ampicillin at 37 degrees Celsius. Transformed colonies would then be screened on LB agar plates containing Xcol and ampicillin. Since the backbone plasmid PUC19 includes the laxate alpha, region colonies with our insert appear white initially and turn blue only in the presence of PFOS. To determine the limit of detection, we could expose transformed biosensor culture to a dilution series of PFOS concentration from 1 nanogram per liter to 10 microgram per liter and record the lowest concentration that consistently result in visible blue color. This informs us on how much sample volume would be needed in the real world testing, allowing us to tailor the biosensor for maximum field sensitivity. So now next part, plasmid design and genetic components will be described by Paris.
So this slide shows the full genetic circuit that we have assembled into the PUC19 plasmid to help build our biosensor. So each part was selected for a specific role to make sure that the system is both functional and easy to implement. So firstly, we start with the J23119 promoter, which is a strong and reliable constitutive promoter from the iGEM registry. It helps ensure that our sensing protein, which is the HLF-ABP protein, is always expressed, so the cell is constantly ready to detect PFOS. The next part of our plasmid is the HLF-ABP protein, which is short for human liver fatty acid binding protein. We chose this protein because it has been shown to bind PFOS with high affinity, which makes it a good intracellular sensor. After the protein, we've got the B0015 terminator, which stops transcription cleanly after HLF-ABP, and it helps prevent interference with downstream parts. After this, we've got the PFOS inducible promoter. So this is currently a placeholder since no natural PFOS sensitive promoter actually exists. So our goal is to replace this with a synthetic ribose switch, which will turn on gene expression only when PFOS is present. So next, we use the B0034 ribosome binding site, which is a standard strong RBS to ensure efficient translation of our output gene. So our output gene is a fusion protein, LPP-OMP-ALACZ. The LPP and OMPA parts guide the enzyme beta-galactosidase to the surface of the cell so that it can interact with excal in the environment. This means that the signal, the blue color, appears outside the cell, making it easy to see. And finally, we end the circuit with another B0015 terminator to neatly close off transcription and prevent leaky expression. Altogether, this design gives us a biosensor that is responsive, visible, and modular, ready to be adapted or deployed depending on the need. The next part will be explained by Priyanka.
Now, let's look at how all the genes in the gene cassette would come together and function at the molecular level as a biosensor. So first, PFOS would enter the E. coli cell through the membrane pores, and inside the cell, the constitutive promoter would drive the expression of our binding protein, which binds to PFOS. And once bound, it activates a PFOS-inducible promoter, which previously explained by Paris that is on a placeholder because it could be synthetically derived as a ribose switch, which can be custom made to get activated to the reaction of binding of our binding protein and the PFOS itself. Once that's activated, it drives the expression of our outgoing gene, which is a fusion protein, has a signal peptide that directs the product bilaxy, that is the beta-galactosidase, to the cell surface, where the enzyme cleaves excal and ends up giving a blue pigment as a visual signal. So all in all, this is a whole cell biosensor, which is compact, autonomous, and easy to interpret, making it ideal for resource-limited settings. In the next slide, Ashish will be explaining how this model of the biosensor would be tested in the real world and deployed in field.
Our biosensor is specifically designed to function outside the lab in the real world, where PFOS contamination is often most dangerous and least monitored. In a field setting, we envision deploying the biosensor using freeze-dried E. coli embedded on paper strips or agar swabs preloaded with excal. These formats are stable at room temperature and can be stored or transported easily. When on site, a user would apply a few drops of environmental water directly onto the stripe or dipstick. If PFOS is present, it diffuses into the rehydrated cells and activates the gene circuit, leading to a visible blue color in under 4 hours. This setup requires no power, no reagents, and no training, making it ideal for low-resource communities or emergency response scenarios. To support deployment, we first determine the limit of detection in the lab, then adjust the sample volume required for reliable detection in the field, typically between 100 microliter to 1 ml. Looking ahead, this same format could be adapted for detecting other toxins or PFAS molecules simply by swapping out the sensing component. With the ability to print biosensor arrays onto the test strips, we see this project laying the foundation for distributed, low-cost water monitoring at a national and even a global scale.